We need to talk about two of the most important concepts when it comes to the nervous system, and that is what we call, well, in actual fact, it's three concepts. It's called a resting membrane potential, a graded potential, and an action potential. Now, they are difficult concepts to get across and to understand, but I think I've honed it in well enough that you should be able to get it just from this one video. So the first thing I want to highlight is this. You can see, or I don't know if you can see it well, but you can see that I've got a person standing there in the anatomical position. However, the head is faced to the side, and you can see the brain, brainstem, and spinal cord inside with a single spinal nerve shooting out and away down the arm, down to the hand. Now, if something were to happen to trigger a receptor in this finger, it could be through touch, pressure, nociception, which is some sort of noxious or potentially damaging stimuli, or whatever it may be. It's stimulating a receptor in that hand. It's going to trigger a signal to be sent down this nerve, up the arm, into the spinal cord, up to the brain, and it will project it to areas that basically control your conscious awareness, that something has happened in that area. So the point of this mini lecture is to highlight what this signal actually is and how it's sent. Generally speaking, people refer to it as you are sending an electrical signal. And they say, oh, we've got so much electricity, we're sending an electrical signal through our body. But that's not entirely true. It's actually an electrical and chemical signal. And we actually refer to that as an electrochemical signal. So all the signals within your body that are sent through your nervous system are called electrochemical signals. What is this referring to? All right, let's shelve that concept for one sec and turn to something called the resting membrane potential, which we actually touched upon when we introduced biochemistry or introduced the chemistry of life. Let's just take a cell, any cell of the body. I told you that outside the cell, we predominantly have sodium. And inside the cell, we predominantly, and let's change the color. Outside the cell, we predominantly have sodium. And inside the cell, we predominantly have potassium, which means there's a concentration gradient, right? So you've got most of the sodium outside, very little sodium inside, most of the potassium inside, but very little of the potassium outside. And there's very specific channels for each of these, what we call ions, to move through. You've got sodium channels, and you've got potassium channels. Now here's the thing. We also have something called a sodium-potassium pump, which is embedded in the walls of these cells. And it's their job to throw out 3 sodium and throw in 2 potassium. So think about that. If you throw outside 3 positive things, right, 1, 2, 3, and throw inside 2 positive things, there is a net positive charge sitting outside the cell. The outside is slightly positive compared to the inside of the cell. But here's the thing, those potassium channels are quite leaky. The lid is cracked open a little bit, and that means potassium likes to go down their concentration gradient. So they like to diffuse. Remember, diffusion is going from a high number of themselves to where there's a low number. So they diffuse from inside, outside, taking that positive charge with it, which even further makes it more positive outside compared to inside. So what you could say is the inside is slightly negative compared to the outside, all because of this process, all right? Now, what we have is that this cell has the potential to change its charge. There's a potential there. Now, this potential to change the charge is called the resting, because nothing's happening, membrane, because there's a charge difference across the membrane, potential. Sometimes written in our books as the RMP, the resting membrane potential. All it is is that there is a charge difference from outside the cell compared to inside due to the movement of those chemicals. Hence why it's an electrical chemical gradient that has been produced. Now this is giving you a hint that we must change this resting membrane potential to send a signal down a neuron. And that's exactly what we do. First thing you need to be aware of here is that when we take a look at the resting membrane potential, we need to have a look at the charges associated with it. So if I were to get something called a voltometer, and what a voltometer does is it actually compares the charge difference from inside a neuron to outside a neuron. Let's draw it up. So if I want to get a little electrode here and a little electrode here and make sure both of them go back to a machine to measure the voltage, what we find is that the inside is negative 70 millivolts. So the inside is negative 70 compared to the outside because the outside has more positive stuff. So what you would be able to draw up on a graph here is if we were to look at negative 90, negative 70, let's just say negative 55, 0, and positive 30, we're sitting here at negative 70. And that's the resting membrane potential. Now, if we want this cell to do something, so in actual fact, it's not just neurons that have a resting membrane potential, but muscles have one as well, and so do glands. Any tissue that has the capacity to do something has a resting membrane potential. We call that excitable tissue. It's excitable. Muscles, neurons, and glands. So let's now move on and take a look at how we can get a nerve or a neuron to send a signal.